Hello friends, today we will learn about Campylobacter, helicobacter, mobiluncus. Here under Campylobacter we'll study about morphology, its culture, biochemical reactions, pathogenesis, laboratory diagnosis, species other than C. jejuni. Next we will study about helicobacter. Under this we will study about Helicobacter pylori, Helicobacter sinadi, Helicobacter fennelli. Next, we will study about mobiluncus. Under this, we will study about morphology, culture, biochemical reactions, pathogenesis, laboratory diagnosis, and treatment. So, let's begin with Campylobacter. Campylobacter are slender, spirally curved gram-negative bacilli. They resemble Vibrous but differ in being microaerophilic, not fermenting sugars and having a lower G plus C content of DNA. There are 10 species of genus Campylobacter, but C. jejuni, C. coli, C. concisus, C. fetus, C. Hyointestinalis, C. lari, and C. sputorum are of medical importance. Now, let's know about the morphology. Campylobacters are small, curved, gram-negative bacilli measuring 0.5 to 5 micrometer into 0.2 to 0.5 micrometer. They are motile by single polar flagellum or amphiterichid flagella. Now let's know about the culture. Growth of Campylobacters occurs under strict microaerophilic conditions. They grow readily on simple media in an atmosphere of a mixture of oxygen, that is 5%, carbon dioxide, that is 10%, and nitrogen 85%. C. jejuni and C. coli and C. lari grow optimally at 42 degrees centigrade. However, C. fetus and most other Campylobacters produce visible growth at 37 degrees centigrade. Campylobacters growing at 42 degrees centigrade are referred to as the thermophilic group. Colonies are circular and convex, but those of thermophilic group, particularly C. jejuni, are flat and tend to swarm on moist agar. Well-formed colonies are seen only after 48 hours. Now let's know about biochemical reactions. All produce oxidase and most species are catalase positive. They are biochemically inactive and do not utilize sugars or produce indole. Most species reduce nitrate to nitrite. Of all the species of Campylobacter, only C. jejuni can hydrolyze sodium hyporate. Now let's know about pathogenesis. C. jejuni has emerged as the most important human pathogen in diarrheal diseases and accounts for 90% of Campylobacter infections. The incubation period varies from 3 to 10 days. All the Campylobacters appear to be inhabitants of the gastrointestinal tracts of animals. Ingestion of raw milk, contaminated water, or partially cooked poultry are common sources of human infections. C. jejuni can produce diarrhea by some mechanisms. Let's know about these mechanisms. It produces a heat labile enterotoxin resembling cholera toxin, in short CT. Second point is, like Shigella and Salmonella, it penetrates gut epithelium. C. jejuni produces a heat labile enterotoxin, in short CJT, and a cytotoxin. These toxins and the invasive property of the organism contribute to the production of damage. The common clinical features are crampy abdominal pain, bloody diarrhea, chills, and fever. The infection is usually self-limiting. Now let's know about laboratory diagnosis. Direct microscopy, gram staining. Gram stained smear of feces may show gram-negative cord bacilli. Now let's know about dark ground or phase contrast microscopy. Wet preparations examined by dark ground or phase contrast microscopy may reveal the darting or tumbling motility of the spiral rods. Direct microscopy may be useful for rapid presumptive diagnosis. Now let's know about the culture. Feces or rectal swabs are platted on selective media. In case of delay in culturing, a transport medium should be employed. Campylobacters survive for 1 to 2 weeks at 4 degrees centigrade in Carrie Blair transport medium. The selective media commonly used are Bursler's selective medium and Campy-BAP selective media. Campy-BAP selective media contains lysed blood agar, vancomycin, polymyxin B, and trimethoprim. Inoculated plates are incubated at 42 degrees centigrade for growth of the thermophilic Campylobacters. If the presence of C. fetus is suspected, additional plates should be incubated at 37 degrees centigrade for its growth. Plates are incubated in an atmosphere of 5% oxygen, 10% carbon dioxide, and 85% nitrogen. After 42 hours of incubation, the well formed identified by gram staining, motility, oxidase test, and biochemical reactions occur. Only C. jejuni hydrolyzes hypurate. Now let's know about serotyping. For epidemiological purposes, C. jejuni and C. coli can be serotyped. Now let's know about the species other than C. jejuni. C. coli. First is C. coli. It causes an infection clinically indistinguishable from that of C. jejuni. C. coli is believed to account for 3 to 5% of Campylobacter diarrheas. It is commonly found in pigs. It can be differentiated from C. jejuni by hypurate hydrolysis test, which is negative in C. coli but positive in C. jejuni. Now let's know about C. conscius. It can grow under aerobic conditions. It has been isolated from cases of gingivitis and periodontal disease. It has also been isolated from pests. Now let's know about C. fetus. C. fetus is a very important veterinary pathogen. It causes abortion in cattle and sheep. It may occasionally cause septicemia in humans who are immunodeficient or have other serious underlying diseases. C. fetus is not thermophilic and grows at 25 degrees centigrade also. Now let's know about C. hyointestinalis. It has been isolated in human patients with diarrhea and homosexual men with proctitis. Now let's know about C. lari. It was formerly known as C. laris. It causes diarrheal diseases, simultaneous C. jejuni infection in humans. It can be distinguished from C. jejuni and C. coli by its resistance to nalidixic acid. Now let's know about sputorum. It is facultative anaerobic. It constitutes a part of normal flora of human oral cavity. It has also been isolated from feces of 2% of healthy people. It may occasionally cause diarrhea and septicemic disease. Now let's know about helicobacter. These are strict microaerophils with a spiral morphology. They possess sheathed flagella. Three species H. pylori, H. synadi, and H. phenili are of medical importance. Now let's know about Helicobacter pylori. In 1983, Warren and Marshall in Australia described a spiral Campylobacter-like organism colonizing human stomach. The organism was initially named Campylobacter pylori and subsequently as Pachy pylori. It has now been assigned to the new genus Helicobacter and called H. pylori. It can be distinguished from Campylobacter by its multiple sheathed flagella and strong hydrolysis of urea. Now let's know about the morphology. Morphology of Helicobacter pylori. H. pylori, Helicobacter pylori, or in short, H. pylori is a short curved spiral or S-shaped gram-negative bacterium measuring 3 micrometer into 0.5 to 0.9 micrometer. It is motile by a tuft of unipolar sheathed flagella, while Campylobacters have a single unsheathed flagella. Now let's know about the culture. It is strict microaerophilic and grows in an atmosphere of 5% oxygen, 10% carbon dioxide, and 85% nitrogen. It does not grow anaerobically. High humidity and 5 to 10% carbon dioxide are required for growth. The optimum temperature for growth is 37 degrees centigrade and not 42 degrees centigrade. It can grow in moist, freshly prepared chocolate, agar, and Campylobacter selective media. After incubation at 37 degrees centigrade under microaerophilic atmosphere for 3 to 5 days, H. pylori produces circular, convex, and translucent colonies. Now let's know about biochemical reactions. H. pylori is biochemically inactive in most conventional tests but produces urease, catalase, and oxidase. It produces abundant urease, which is 100 times more powerful than that produced by P.R. vulgaris. It turns Christensen's urea broth to pink color in a few minutes. Now let's know about the pathogenesis. The only known natural reservoir of H. pylori is human gastric mucosa. Gastric antrum is the most favored site, but any part of the stomach may be colonized. The organisms are present in large number of mucous, overlying mucosa, and also deep in gastric glands. H. pylori is implicated to cause chronic active gastritis, duodenitis, and peptic ulcer. It is also recognized. It is also recognized as a risk factor for gastric malignancies, namely adenocarcinoma and mucosa-associated lymphoid tissues. In short, MALT lymphomas. The virulence of H. pylori has been associated with certain alleles in genes such as CAG, that is cytoxin associated gene, and VAC, that is vacuolating cytotoxin gene. Now let's know about laboratory diagnosis. Specimen biopsy of gastric mucosa, direct microscopy. Gram staining. Gram staining of the specimen shows short curved spiral gram negative bacteria, hematoxylin eosin staining. It can be used in histological sections, Warthin sterile silver staining. Organisms can be seen most clearly with this method. Now let's know about the culture. Specimen is inoculated on chocolate agar and Campylobacter selective medium followed by incubation as mentioned. As we studied till now, the organism is identified on the basis of colony morphology, gram staining, and biochemical reactions. Now, let's know about the biopsy urease test. The biopsy material is placed in urea solution which as indicator and incubated at 37 degrees centigrade. If the tissue contains H. pylori, pH changes with a few minutes to 2 hours due to production of ammonia. Urea is broken down to ammonia by action of enzyme urease produced by the organism. Now let's know about the urea breath test. Urea labeled with an isotope of carbon is given orally to the patient. If the patient's stomach contains H. pylori, urea is converted into ammonia and carbon dioxide. This labeled carbon dioxide appears in the breath where it can be measured. Now let's know about the serology. Antibodies to H. pylori can be detected in patient serum by ELISA. Now let's know about the treatment. Treatment is indicated only for H. pylori associated gastric or duodenal ulcer and not for asymptomatic colonization. A combination of bismuth, salicylate, tetracycline or amoxicillin and metronidazole are given for 2 weeks. A combination of imeprazole and clarithromycin is another alternative. Now let's know about the helicobacter cinnaei. It was formerly known as Campylobacter cinnaei. It has been isolated from rectal swab of homosexual men with symptoms of proctitis. It has also been described as a cause of bacteremia in homosexual men in HIV positive individuals. Now let's know about Helicobacter phenoli. It was formerly known as Campylobacter phenoli. It has been isolated from rectal swabs of homosexual men. Now let's know about mobiluncus. Mobiluncus is the generic name given by Spingel and Robert in 1984 to a group of bacteria isolated from the human vagina with bacterial vaginosis. Recent studies suggest that genus Mobiluncus belongs to the order Actinomycetes and it is closely related to the genus Actinomyces. This genus has 2 species, namely M. cutisi and M. mulieris. Now let's know about the morphology. Both species of Mobiluncus are slightly curved bacilli and occur singly or in pairs. M. cutisi is short, measuring 1.7 micrometer into 0.5 micrometer and gram variable, while M. mulieris is long, 2.9 micrometer into 0.5 micrometer and gram negative. Both species are motile by means of subpolar flagella. Now let's know about the culture. They are essentially anaerobes but can grow in the presence of 5% oxygen and 95% N2. They can be grown on blood agar medium at 33 to 37 degrees centigrade under anaerobic conditions. After 5 days of incubation, round, low convex, smooth, translucent, colorless colonies 2 to 3 mm in diameter are produced. Now let's know about the biochemical reactions. They are oxidase and catalase negative. And now let's know about the Pathogenesis. Mobiluncus species have been found in bacterial vaginosis in association with bacteroids species and Gardnerella vaginalis. Bacteroid species constitute a major component. There is a foul-smelling, non-purulent vaginal discharge. Now, let's know about the laboratory diagnosis. Microscopic examination of fresh unstained smears of vaginal discharge show epithelial cells coated with organisms. Mobiluncus species may be isolated by inoculation on blood agar and incubating and anaerobically. Colony morphology, gram staining, and biochemical reactions can be differentiated into two species. Now let's know about the treatment. Penicillins are used for the treatment of infections with mobiluncus. Organisms are also susceptible to clindamycin, erythromycin, and gentamicin. Finally finished. Any questions regarding this? You can ask me in the comment box and subscribe my channel to know more updates. Thank you.